Hello, I'm Dr. Stephen Pipe from the University of Michigan. My co-authors are presenting a oral presentation at ASH this year on Etranacogene Dezaparvovec. This is an AAV5 capsid vector technology for gene transfer in adults with severe or moderate severe hemophilia B, or factor IX deficiency. This vector includes a hyperactive form of factor IX called the Padua variant, which boosts the activity potency of factor IX by about 6 to 8 fold. And what we're reporting at this meeting is now the 2-year follow-up data from this phase 2b trial. We had some insights on this AAV5 vector from a previous version, which included a native factor IX transgene. And those patients are also being presented at this meeting, and they've had stable expression of factor IX now for up to 5 years with effective reductions in annualized bleed rate and low requirements for factor IX replacement with no late emergent safety signals. So the purpose of this phase 2b study was to take the safety and efficacy observed with that vector and simply modifying it by using this hyperactive factor IX Padua variant. So there were 3 subjects who were included in this trial. And again, based on insights from the previous trial, we learned that pre-existing neutralizing antibodies against this vector did not seem to have any impact on the efficiency of transduction with this vector. So for the phase 2b study, we did measure the pre-existing neutralizing antibodies against the AAV5, but they were not excluded from participation. So the 3 gentlemen who participated, they were ages between 43 and 50. They all had factor IX levels of less than or equal to 1%, and they had all been on extended half-life factor IX prophylactic therapy coming into the trial. And each one of these did have a positive titer against AAV5. The primary endpoint of the study was the factor IX activity of at least 5% or higher at 6 weeks post-transduction. And this was achieved in all 3 participants. What we know now is out through 2 years of follow-up, the mean factor IX activity in these 3 individuals has been 44.2%. 2 of the participants are now in the non-hemophilic range, so above 40%, and one's in the high mild range at 36.3%. All the participants remain prophylaxis-free through 2 years of post-treatment. And from a liver-specific safety evaluation, no clinically significant elevations in ALT or AST, no evidence of AAV5-specific T-cell responses, no requirement for immunosuppression, and no loss of factor IX activity over time. So overall, this therapy looks like it's well tolerated in these 3 patients with no new treatment-related adverse events now out through year 2. And all the participants have achieved sustained factor IX activity in a functionally curative range after this single administration. The participants are free from bleeding, and they're not having to use factor IX replacement therapy on a regular basis. So this it really is giving good impetus to evaluate the full cohort of patients who are being studied in the phase 3 HOPE-B study, which is also being presented as a late-breaking abstract at this meeting.